My name is Ryan Sullivan. I am one of the medical oncologists that takes care of melanoma patients. And today I have the pleasure of introducing Chloe Villani and Carrie Reynolds, who are going to talk to us about severe immunotherapy complications and integrating clinical insight with translational innovation. A very brief history. The first immune checkpoint inhibitor to be approved by the FDA was in 2011. It was followed soon there. That was ipilimumab in melanoma, followed a few years later by anti-PD-1 antibodies, pembrolizumab and nivolumab in 2014. And throughout the decade, essentially, of the teens and a little bit in the aughts, we began to realize that these drugs can cause extreme side effects. And that we were getting really, really good at causing side effects, but we're not really, really good at treating side effects or figuring out why they were happening. And thankfully, Carrie and Chloe sort of came together in a moment of sort of panic for those of us that were caring for these patients, but brilliance for our institution and really the field writ large, because their collaborative relationship has essentially changed the way we think about managing these problems. So it is my distinct pleasure to introduce Carrie Reynolds. She is the director of our Severe Immunotherapy Complications Service. You can stand up if you want. It's okay. She's also the clinical director of the inpatient cancer service. And then she's going to get started, and then Chloe's going to join her. Chloe Villani is the director of the Single Cell Genomics Research Program at the Krantz Cancer Center, or Clinical Center, no, sorry, Center for Cancer Research, sorry, everybody, and also the Center for Immunology and Inflammatory Diseases. They're both assistant professors at Harvard Medical School, and it is a great pleasure to introduce you.
Thank you so much, Ryan. And it's really a pleasure to be here to be able to present on behalf of an entire team. So I have to tell you where it all started. So this is David. He was a father, he was a husband, he was a CEO, and he was also one of our first patients with metastatic melanoma to be treated with 2 immune checkpoint inhibitors, ipilimumab as well as nivolumab. He did well with cycle 1, but after cycle 2, within just a few days, he presented to the Mass General Hospital emergency room with an oxygenation of 48%. That admission, he was diagnosed with pneumonitis, or inflammation of the lungs related to the dual immune checkpoint inhibitors. At the beginning, he did very well with steroids. He got out of the hospital. But 4 weeks later, he represented with relentless immune activation attacking his gut, causing colitis, as well as his neurological system. We tried everything. And a few days before he was headed to the ICU, he actually looked up and he said, "Doc, I know this isn't good. Please tell me you will learn from this." And we promised David on that day that we would learn from this. And it's the collective you here. And we promised him every day since. He was our first autopsy. In the time of autopsy, there was not one single S100 positive melanoma cell. The disease was eradicated from both his lungs and his brain. And so it truly embodies immune checkpoint inhibition, because it is such a promise, but there are perils that we have to understand. And that's when we reorganized here to think about this differently. And just because the Cancer Institute now has a wide variety, a diverse audience, I wanted to give a little background on the landscape of immune checkpoint inhibitors. So Immunology 101, many times a day, your body's trying to decide between self and non-self. And right here, this little blue ball is an antigen. So an antigen is presented on a major histocompatibility complex molecule of an antigen-presenting cell like a dendritic cell. And it's presented to the T cell receptor on this bluish purplish T cell, and that is signal 1. But that alone does not activate the immune system. You must have a co-stimulatory signal, that second signal of CD28 and CD80 or 86. Then the T cell can be off to the races. It secretes cytokines, it proliferates, it migrates to the tissue. But with immune activation, immediately checkpoints come into play so that we can regulate that immune activation. And a checkpoint such as CTLA4 comes to the cell surface and it can outcompete CD28 for that co-stimulatory action and actually be able to decrease the immune activation. Similarly, in the periphery, the T cell actually expresses programmed cell death 1 or PD1, and it can meet a ligand in the tissue, PDL1 or PDL2. And again, you see that this signal decreases immune activation, sends the cell into quiescence. However, cancer evades the immune system. And so monoclonal antibodies designated to these checkpoints have been developed. There are now 15 that are approved. You can see the majority are an anti-PD1 and anti-PD1. And these agents are truly bending the survival curve. I like the long-term data. So this is one of the longer-term studies, 6.5 years out of one of the early melanoma studies that looked at the overall survival of ipilimumab. That first drug is in green, showing 23% of people live. In blue is the PD1, 42%, or combination is 49%. To put this into perspective, if David was diagnosed in 2005, before these were approved, the overall survival for very significant disease to the brain was 4 months. So this is truly remarkable to see these long-term survivors. And at ASCO just a couple months ago, I loved the final analysis of the phase 3 checkpoint 214. Over 9 years of follow-up data, looking at immunotherapy and intermediate or poor risk renal cell versus Sutent, which is a tyrosine kinase inhibitor. First line therapy. And if you look at those overall survival curves with immunotherapy, 46.7 months versus 26 months with a hazard ratio of 0.69. The progression-free survival hazard ratio, 0.7. And the durable responses, 82 months versus 19. So truly incredible. Each of these boxes is an approved FDA indication for immune checkpoint inhibitors. Just like Ryan said, the first was 2011. We lost David in 2016. And for several months after, we thought about how do we reorganize? And we started the severe immunotherapy complications effort October 3rd, 2017. Since that time, now there are 113 FDA approved indications as of March of this year. And the dark blue boxes are where there's combination approvals. The red outline boxes are where we're moving it to adjuvant or neoadjuvant. So it is clearly moving to early stage disease. And if we look at our own institution, how much immune checkpoint inhibitor are we using? If we look at the patients in our registry until June 9th of this year, you can see it's increasing year by year. Each color is a different immunotherapy regimen. And in Boston, we've treated over 11,000 patients. Last year alone, we treated 1,474 patients, and we've already treated in the first 5 months 1,000 patients. So with these indications, the use is clearly rising. We admitted about 320 of them for suspected serious immune-related adverse event. Then it's not just downtown. This is also in the network. This is MGH satellite and affiliate, and you can see the tsunami of the straight up line. So it is here to stay. It is no longer just metastatic melanoma like treating for David. Instead, this looks at how much each of the disease centers are using immune checkpoint inhibitor. And now the highest use is in thoracic, followed by GI, followed by GU cancers. With this promise does come a wide range of autoinflammatory-like toxicity that can affect nearly every organ system in the body. Not at the same time. Some only get one, some get multiple like David. We talked about how it affected for him the lungs, the colon, as well as the nervous system, but this can affect the heart, the liver, kidneys, and so on. How often does it happen? Well, any toxicity can occur over 70% of the time, but most are very well managed and easy to manage in the outpatient clinic. Severe toxicity, though, with single agent PD1 can happen 14% of the time, or with the older CTLA4 agent, 34% of the time. Or if we're combining these two agents, it can happen, depending on the dose, upwards of 55%. So serious toxicity has to be considered in these patients. You can see that column where patients had to stop treatment because of toxicity. Thankfully, death is rare, only about 0.5 to just over 1%. But as we treat hundreds of thousands of patients across the United States, these numbers are incredibly important. Our colleague here at MGH, Eugene Semenov, along with our colleague at the Brigham and Women's, Nicole LeBlanc, recently put an article in Lancet Oncology looking at the incidence in the MGB system, as well as a Trinetics database. GI is most frequent for toxicity, 23%, if you add on that plus hepatitis, which is 18%. Endocrine can happen 36% of the time, cutaneous 24% of the time, musculoskeletal 23%. Now, some of these are good signs prognostically. So in their study, they found associated with overall survival were endocrine and cutaneous, but some of these are detrimental. So things like respiratory or neurological IREs were associated with worse survival. And it was 2017 where we brought this wheel together. And now it's over 60 members across 6 departments and 10 divisions of medicine. And the key was to have subspecialists that could really handle all of the different organ systems. We do exactly what Dave Ryan sets every program out to do. 1, take the very best care of patients that you can. 2, move the field forward. And 3, teach the next generation while doing it. And so clinically, we care for patients. We have over 29,000 patients in our registry, and over 2,000 samples have been collected, which Dr. Chloe Villani, who leads the translational effort, will tell you more about. Clinically, when we care for these patients, they can be inpatient. So we have the severe immunotherapy complication service, which is an oncology service that sees these patients. We actually started that service with an EPIC report that helps us find out where these patients might be. And the same infrastructure exists at the Brigham. So 2 years later, we were able to start up the ITOX service over at the Brigham. So it's similar on both sides. We looked at our severe immunotherapy complication service, and when we instituted, the readmission rate for IRAE went from 26% to 15%. So a significant decrease. And if they were readmitted, the length of stay dropped by a day. So we published that work. In addition, we have urgent immune-related adverse event clinics across these subspecialties that fit our patients in. And because we're a shared network in the same electronic medical record, we're actually able to use EPIC to contact and to work with all of our hospitals like Danvers, as well as Newton Wellesley and on the islands. I want to point out that this is substantial integration of sample collection. And the reason why we're able to do that in this effort is because of what already exists at MGB. So first, I just want to point out all of the important people in 11181, the melanoma biobank that has been critical to both support and allow us to collect under. And then Dan Urich with 13416. This is a broad cancer center protocol that allows to collect across different diseases, as well as integrates a rapid autopsy program. And now I'm going to be bold, slightly controversial, and at the risk of kind of shocking maybe over 90% of the Cancer Institute that might be on the call. But every cancer center everywhere has medical oncology, radiation oncology, and surgical oncology. What sets ourselves apart and what the MGB Cancer Institute will be able to do when we think of caring for patients will exist because of the subspecialty expertise that exists on the wheel at the top, which is MGH, and the wheel on the bottom, which is the Brigham. It is truly those individuals in these divisions of medicine with the expertise of drug toxicity, of autoimmunity, of procedures. This is why we can care for patients differently, and we will care for them standard of care on the left, as well as integrate into experimental therapeutics. By bridging the Mass General and the Brigham, there is nothing else like it anywhere else, and it is not able to be replicated. And I want to also show to you that this has been a collaboration across town that has existed for years, far before integration or institute were words that we were thinking about on a daily basis. So I'm going to just highlight 2 examples because I don't have the time to show you all the spokes on the wheel, but I wish I did. But cardio-oncology, we have Dr. Tom Nealon on this side. We have Dr. Anju Noria, both cardio-oncology world experts that have come together since 2018. And these are the key projects they've done together. They've also trained the next generation with Dr. Dan Zotov, who is now faculty, as well as Dr. Raymond has just joined, who's interested in immunology and endovascular biology, which will be incredible. Same thing with onconephrology. We have Dr. Seiss on this side. We have Dr. David Leaf at the Brigham. They've trained the next generation, 2 of which are already faculty, and 1 is a fellow. They have dated this back to 2018, where they have described and phenotyped nephritis. In addition, really shown us predictors And all of this work has highlighted the gaps that exist because we do not have diagnostics in this field. We do not have validated biomarkers. And I want to bring this back to a case. What does this look like on a day-to-day basis? So this is a 70-year-old female breast cancer that we saw. She had high tumor mutational burden. And at the time, high TMB was not treated with immunotherapy. Now we know that advanced cancer with high TMB or high microsatellite instability are actually eligible for immune checkpoint inhibitor, but she was on trial. She had a history of hypothyroidism, so some autoimmunity already at baseline, and she was given 2 immunotherapies, and she had 2 doses similar to David. She came in and she had double vision and she had eyelid droop. That clinically is very concerning. For is there something with the nerve that is innervating the muscle? Is there inflammation in that neuromuscular junction? In addition, it was a concern for hepatitis with LFT elevation and some mucositis. In the ED, many of the basics were done. Her EKG did show new AFib, which triggered another thought in us, and labs were sent. Initially, the plan was to send her to the ED for a respiratory evaluation. If that looked okay, she was going to have an EMG outpatient. So the workup of these patients, what we have learned over time is these conditions can overlap. And the neuromuscular can overlap with muscle inflammation, myositis, as well as myocarditis, with this inflammation of the heart. Her CK was normal at 117, so did not look like myositis. Her exam, though, when she did her exam, we could tell that she had right ptosis. She had impaired upward gaze bilaterally. She had moderate weakness when it came to trying to close her eyes, and she had extremity weakness 4 out of 5. Unlike myasthenia that presents in, say, neuroclinic de novo, this is often antibody negative, just as it was in her case. In addition, the EMG was not even helpful. It was normal. We had respiratory therapy come and see her, and this is critical for anyone you suspect of myasthenia because of the fact that the phrenic nerve innervates the diaphragm, and there can be inflammation, and you need the diaphragm to drop so that you can actually have respiratory function intact. Without it, you can go into respiratory compromise quite quickly. So we had that respiratory therapist come to check her negative inspiratory force and force vital capacity. Thankfully, both were normal. Anybody that presents with neurological findings like this, a brain MRI when they have metastatic disease is critical, and hers was negative. Clinically, she has myasthenia. We are going to treat the myasthenia, but just to give you a sense of how little diagnostic help we had. In addition, with that experience, we knew it was important to send a troponin before we ever sent her home. And so she had an elevated troponin to over 102. That's over 10 times her baseline, which was normal. Her EKG, as I showed you, or as I told you, had AFib. But when we look at evaluating for myocarditis, her echo was normal. Her cardiac MRI, normal. There was no late gadolinium enhancement for her. And when we think about these patients, the issue is her suspicion was still so high because she already had 1 IRE, she had an elevated troponin and AFib, that we took her to a biopsy of her heart, of the right ventricle, while the heart's still beating. And that was really the diagnostic test that we needed in terms of showing active myocarditis. She was treated with steroids, and because cardiac and neurotoxicity are the most fatal, we use often high dose or pulse dose steroids, and that's exactly what she had before switching over to prednisone. I just want to give you a sense from what Tom and Andrew and Dan have really added to the field. When we think of that initial case series of their 35 patients, they showed us that troponin is often elevated in this condition. Myocarditis happens in about 1% of cases. But in this study, 46% of the patients had a major adverse cardiac event. What MACE means is cardiac arrest, death, shock, ventricular rhythm, or heart block causing hemodynamic compromise. In addition, EKG can be somewhat helpful. Many of those are subtle findings, but the EF, so the ejection fraction, 49% only have an abnormal echo, but many are like our patient that have a normal echo. Dan Zlotov then took all of the EKGs and really looking at 140 myocarditis patients from across her institutions, as well as a registry, and showed us that QRS duration on the EKG is very important and associated with myocarditis. In fact, for every 10 seconds you get that are prolonged, 1-point-fold increase in that major adverse cardiac event rate. They went on to help us with imaging characteristics because EF is not helpful on echo, but they were able to show that a new parameter, global longitudinal strain, was helpful. And decreased GLS is associated with myocarditis. And in fact, for every percentage that decrease global longitudinal strain, you have a 1.5-fold increase in those very serious outcomes of MACE. They also showed us what we saw in our case, but the cardiac MRI is a gold standard imaging technique. But out of 103 of their myocarditis patients, only 48% had true late gadolinium enhancement. That is a substantial number of false negatives. Now, if you drop your EF, that can go up to 55%, but it just goes to show you that our image modality is not enough. Now, our patient actually donated blood and tissue to the translational effort. How did she do? Well, her first troponin is down below at 102. She was treated with steroids and she did well initially with her troponin falling, but then her troponin went back up because she was refractory. So we started CellCept, 1 gram BID. It took 6 months to wean her steroids. It took over a year to wean her CellCept. It was 1,275 days to get to a totally normal troponin. I hope I came across with how hard this can be diagnostically. And so the goal of the translational research effort from the very beginning, when it started just a couple months after the effort, what immune cells and gene activity are present in heart tissue and the patients with myocarditis? What are these cells recognizing? Are they the same culprits that are found in the tumor, the heart, and the blood? Is there anything to detect this earlier? And can we figure out new treatments for myocarditis, which are sorely needed? I'll turn it over to my colleague, Dr. Vellani.
Oh, she's going to be back. She's going to be back. Well, you can clap again. And so answering these biological questions is really at the foundation of ultimately finding better solution to mitigate and improve patient care, right? Right now, we actually have really bad biomarkers to predict who's at risk. We don't have good diagnostic tools. These invasive biopsies, while we can do them in the MGB system, not so easy to implement in community settings. We also need better targeted therapy solutions. To give you the example of this patient, using steroids can work, but can also impact your anti-tumor immune response. So double-edged sword. And so this is obviously overly simplified, but some of the solutions can involve withholding, stopping your life-saving immunotherapy, or using these steroids. And on the research side, preclinical models which were used to identify the target of these amazing drugs, these models don't develop toxicity aside from vitiligo. So there's actually a need to study these presentations in patient specimens, which is what we're doing. And so better biomarker, better treatment solutions starts by understanding the biology. So I know we're shifting from pure clinical presentation to pure biology. I'm going to try to not traumatize the entire audience. I'm going to try to keep it as high level as I can. But it's not a simple one-size-fits-all solution. It's not a single mechanism, which makes it so hard to think about therapeutic solutions. So I'll give you some examples, high level, of some of the current hypotheses. Can you see my cursor? Yes. So it could be what we call cross-antigen reactivity. What does it mean? For example, melanoma. Some melanoma patient will develop vitiligo. The immune cells come in and start killing your melanocytes. By killing melanocytes, you're going to have bystander cell killing, and you can trigger cross-antigen reactivity like vitiligo. You can have an off-target effect. Your pituitary gland express the drug target of anti-CTLA-4. And so you can get hypophysitis. However, you don't get the same type of hypophysitis in response to anti-PD-1. When you give checkpoint, you're diversifying the repertoires of T and B cells in your body, right? And as you're diversifying, you can give rise to autoreactive T and B cells that starts attacking cells. You might also be somebody who was at risk of developing autoimmune disease all your life. You had some autoantibodies, but at sub level in circulation, that was a trigger to bump it up. That can trigger autoimmune reactions. There are some studies that have shown that microbiome can play a role in shaping your response. We already know that some cytokines and chemokines might be tied to adverse events. You have these amazing cells that are called tissue resident memory cells, these T cells in your tissue. They represent 80% of the T cells in your different tissue. They're there to do recall to pathogens, to develop a quick response when you have any type of triggers. It's been shown, we've shown it at MGB, that they're involved in the context of colitis. It's been shown that it's also involved in some forms of pneumonitis and also skin tox. There's been some beautiful study also from Boston, where our sites were involved, showing that some patients are genetically predisposed, but it's so much more complex because these drug targets are expressed on many cells beyond T cells. So how do we go about this? And so this is already outdated because we keep on collecting over the last few years. Of course, we slowed down during the COVID pandemic, but we've collected well over 500 samples from 500 patients across all different talks. This is in the context of center of care. It's not clinical trial. And so you can imagine the work that entails incorporating this whole system as part of patient care so that we don't slow down patient care. It's an amazing teamwork. You can see we have case and control. Everybody's a cancer patient. Everybody is receiving one of these checkpoints. You need to control for patient receiving checkpoint because receiving checkpoint does change your immune system, right? And so when we collect the samples, when Carrie's team contacts us, we actually don't know if it's going to be a case or control, but we proceed. We have to then wait for the pathology reports to know if this was indeed a case or control based on immune infiltrates. So we've collected well over 2,000 specimens because we collect, as you're going to see, the tissue, the blood, the body fluids, and so forth. We are trying to define these biomarkers and drug targets by exploring scale, time, modalities. What do I mean by that? Yeah, I mean going from organs to genes, right? I want to know if the biology attacking your liver, your heart, your brain, your lung, is it the same? Should you be treating all these patients the same way? Do we need to tailor it to every organ system? So how do we go about this? So when we can, we have biopsies of these organs. I'm going to show you some of the technologies we use to dissociate the tissue and try to kind of analyze every single cell. And within the cell, we try to figure out the molecular network to see if it's a molecular network that's pathogenic or not. That's scale. Time. When you do human studies, unlike mouse, time is your best friend. Time points, because you can track over time what tracks with the evolution and resolution of disease. That can give you a hint at what really matters in terms of tracking. So what we do, we collect event trigger specimens. So when there's a suspicion for IREs, we collect before any steroids, blood serum, plasma, biopsy when we can. And what do I mean by relevant body fluid? If it's a kidney, we also collect the urine. If it's the brain, the CSF, the lung, the lavage, the joint, the synovial fluid, because sometimes these fluids are a little easier to collect. And what if we could find some factors in these fluids that could become diagnostics, right? So that's part of the collection. And then we collect until resolution or relapse, post-steroids, because we want to see what factors disappear after they get better or not if they're refractory. And Carrie mentioned 11-1. The team has been an amazing partner in the context of cutaneous melanoma. They actually collect also as part of center of care. And so that gives us an opportunity to go back in time for the samples that are paired and see what was there before starting immunotherapy and before IREs. Modalities. So we use a lot of cutting edge technologies to try to answer these questions. I'm going to tell you a bit more about single cell multiomics, but we layer information to try to draw the bigger picture of what's happening. So before, I want to make sure that I don't lose everybody when I talk about single cell genomics. So I'm going to use my favorite analogy, a fruit salad. So the fruit salad would be the equivalent of looking at all the cells in a piece of tissue on a biopsy, right? And the fruit salad, you would have different types of fruits, same thing, different types of cells. Historically, the way we've been analyzing the content of your biopsy is the equivalent of taking all these fruits and making a fruit smoothie. You're making the average. And so when you do the average, some of the perhaps rotten fruits would get lost in the fruit smoothie if you only had a few rotten fruits. The rotten fruit here would be the analogy of pathogenic cells. And so now we have these new technologies. That was partially how I was recruited to MGH. And you can see how this connects, because I started in October 2017 and the sixth service started in October 3rd of 2017. I brought in all of these single cell genomics technology with me. Single cell genomics allows you to pick every cell apart one at a time. We take the single cell suspension. We have different ways of capturing the cells one at a time, bursting them open and extracting the genomic information. That's the equivalent of taking the bowl of fruits, flipping it on the table, and then organizing every fruit by types of fruits and figuring out the quantity and the content. And you could go even deeper in my analogy. Like you could say I have apples, I could tell you what type of apples or what type of T cells you have. And so that's a lot of what we do. And so how do we go about answering these questions that Carrie listed for her case, right? And so I'm gonna walk you through the strategy and also show you examples. So we have three main pillars that we're using. The first one, and they're all geared towards defining biomarkers and drug targets. The first one focus on the tissue. This is one of our major strengths at MGB, actually, because there's a lot of other groups out there throughout the United States that are mostly studying blood because it's easier to access. And of course, you may find pathogenic cells in blood, but it's like looking for a needle in a haystack. Where these pathogenic cells are most enriched is in your tissue. And so we always start with the tissue here. I'm going to show you how we do this and trying to define potential drug targets through the lens of defining pathogenic cells. Then we want to try to understand the relationship between the IRE, immune-related adverse events lesion, the tumor, and the blood. Why is that? In many cases, people always assume that your toxicity is tied to your tumor response and you cannot decouple it. But there are many instances where it may not be. How can you figure this out? The T cell receptor I was going to show you is like a unique barcode. So if you can look for this barcode across compartment, you can figure out if you're looking at the same thing. If we can decouple, it could mean that we could find drug targets that could allow us to keep your patient on their life-saving therapies while mitigating the toxicity. The last one is about biomarker. Anything we find in the tissue, we want to see if it's in the blood. So let's go through an example. I'm going to show you the myocarditis study design with lots of co-first and co-last, which really kind of captures the essence that it takes a village to do this. And so we analyzed heart tissues from 8 controls, from 13 cases on myocarditis, 7 that were on monotherapy, 6 on combination therapy across 8 different tumor types. Through the 13416 protocol with Dr. Urich, we actually had access to paired tumor specimens collected at time of autopsies with the heart. And we had paired blood specimens for all of our heart. We also had additional cases where we didn't have access to heart, but we actually had access to extra blood. I'm going to show you we did some single cell RNA sequencing, which we're not going to go through. We did microscopy imaging of the heart. We looked at secreted factors. For every analysis I'm going to show you, we're going to be comparing data from control heart and blood. The control hearts come from either patients that did not have checkpoint exposure or were on checkpoint and didn't have myocarditis, to patients with myocarditis that were diagnosed by MRI or histology. And we're going to try to define the cell population, the gene programs, the T cell receptor, and the circulating factors that are enriched in these cases. So we're going to start with the strategy number 1. In the heart, what are the cells and molecular perturbation that defines the tissue? Okay, so the way you're going to see this map is after I extract every little cells, extract their genomic features, we sequence them, we can use that information to create unique identity for every cell and try to see the cells that have the same identity. And so here represented by color and shape. And I'm going to compare control to disease, and all the little color points you're going to see are going to be indicative of a cellular population. And I'm going to try to see the cells that are unique in disease, like the pink ones and the green ones, for example. So it allows me to figure out what are the guilty cells driving the disease, and if it's only mediated by T cells, which, as Carrie introduced in her 101 overview slides, are key in modulating the drug response. Beyond that, let's say I find this is a pathogenic cells, I can try to understand if the cells is a healthy cells or pathogenic cells are part of a spectrum. I can predict who they talk to in the tissue, what are their neighbors. I can see how the cells is wired molecularly, because maybe that's the way I can reverse the pathogenicity. And I'm also interested in understanding the relationship to autoimmune disease. So let's start with the heart. What did we found in the heart? Every little dot here is a cell. The color tells you the groups of cells with share identity. And the arrows tells you if it was up or down in myocarditis. Unsurprisingly, you're not supposed to have many immune cells in the heart. So we see a lot of myeloid cells, T cells, and so forth. But that's one level of abstraction. You can zoom in on a particular group of cells and ask, are there different flavors of T cells? Here we find 6 different flavors of T cells. And I can tell you what the little dot cells express. Some are ready to kill. Some are exhausted, expressing PD-1 and LAG-3. Now you can ask, are any of these population more present in myocarditis than in control? In red is myocarditis, in gray is control, every dot is a patient. And you can see almost all of them are more abundant in cases in control, and those are not. It's really more a power issue. Now, it's not surprising to see T cells in the heart. That's actually how we can diagnose myocarditis, but we didn't expect that many flavors of T cells. There are many different flavors of it. So we ask, are these the same T cells fighting the tumor, right? Trying to decouple this, which brings us to the second strategy. So I told you about every T cell is a T cell, which is like a barcode. So you can ask, can I find the same T cell across all compartments? And can I find TCRs that are shared across patients with the same toxicity in our bigger program across different toxicity? Oh, we have Dr. Blum at the back, who's the lead author of the study that I didn't see, putting him on the spot. And so, and of course, once we figure out if these TCR clones are shared, you can try to figure out if they're recognizing the same antigens. And so this is where 13-for-1-6 partnership has been so essential. We were able to collect paired heart and tumor tissues and adjacent control tissue, which is really important to identify these bystander TCRs that may recognize this different viruses. Working with pathologists here at MGH, we were able to do serial section, do some H&E staining, and identify the areas of active inflammation and normal parenchyma. And then the team scraped the tissue and isoHDNA, we did the T cell receptor sequencing. Now you have to know that before this study, the gospel was that for myocarditis, these TCRs were shared. It was an elegant study in the New England Journal of Medicine. We actually show that it's not the case, at least not in every patient. What I'm showing you here is every dot is a different TCRs. In green are those that are expanded in the heart, in purple in the tumor, in red those that are shared. So if you take home message, almost none of them were shared, and some are extremely expanded in the heart and very expanded in the tumor, and they're not shared. So what do they recognize? Because that could be a way of sensitizing the patients. There was a beautiful study published a couple of years ago in Nature that suggested that alpha-myosin was the key antigen. And so what we did is every row, every column here is a patient. We selected the top 70 most expanded T cell receptor along with controls, including some from this study. And you can see some are really expanded, like over 40%. Then we clone these T cells, we express them in donor T cells, and we expose them to these antigens, including alpha-myosin, but also troponin. This was an obvious candidate. And if the T cells get activated, which we can tell by doing measurements of CD137, which is a marker for activation by FACS, then you know it's recognizing these antigens. And this very blank heat map tells you that none of our TCRs recognize alpha-myosin or troponin. The only ones that did were these controls TCRs from the original study. So the assay worked, but what we're putting forward is that there's other antigens involved, which is something we're following up. Finally, the third strategy I told you about is about identifying biomarkers for patient at risk. So for anything we find in the tissue, we want to see if it's in the heart, in the blood. Could be a secret factor, could be a cell, could be a TCRs that we could track over time. We are also interested in understanding if there's specific secreted factors or autoantibodies that track with evolution and resolution of disease. So I told you that there was almost no TCR sharing between the heart and the tumor. But were the expanded TCRs in the heart present in the blood was our next question. So we sequenced the blood of these patients. Every little dots here is a cell. Every color is a cell subset we have defined. We took all the TCRs we identified in the heart because they're barcodes, right? And we look if these barcodes were in the blood. And when you do so, you can find these TCRs in the blood. This is a composite of all patients combined together. What was interesting is when we look at the results one patient at a time. It turns out that the patient who died of myocarditis shared their TCRs with a particular cell subset, and those that didn't die shared with another subset. And so imagine, like, there might be a possibility of identifying the blood who may die of myocarditis. And you can ask, what's so special about the cells that are in red, which we call this cluster 12? These are cycling by this marker, CD8 T cells that express a specific chemokine receptor, CXCR3. So we ask, CXCR3 recognize specific ligands like CXCL9, 10, 11. So we ask, can you detect the protein in the blood of these patients? So we did Luminex here. Every dot is a patient, red is myocarditis. And indeed, we could identify CXCL9 and 10 in the blood of these patients. So let this sink in for a moment. It means that we might have identified some potential targets in the blood of these patients that could predict fatality. Obviously, that needs validation in a much larger cohort, but it opens all new possibilities. We also identified several additional candidates that require further validations. There's a lot of dynamics happening in the blood. Obviously, we want to understand if these factors could also help us distinguish response to toxicity. And this is tricky because these factors are also tied to tumor response. And we also want to see how early we can detect them. Beyond T cells, we also showed that myeloid cells, here we define 6 different subsets, are also up in myocarditis, here in red. And interestingly, the same one, including dendritic cells, that are up in the heart go down in the blood, which suggests trafficking. An even more interesting observation is that we showed that the dendritic cell abundance in the heart was associated with elevated troponin T. Here, as a reminder, when the heart muscle is damaged, troponin T leaks into the bloodstream. It can be a marker of severity. And so it suggests that the more dendritic cells you have, the more severe disease you have. And we actually could find these dendritic cells in the heart talking to T cells, more in cases than in controls, validating this. Beyond myeloid cells and T cells, we also looked at all of the non-immune cells. And what really caught our attentions were fibroblasts. Fibroblasts, which are key structural cells, were up in the heart of patients. And when you zoom in on fibroblasts, we found 6 different flavors of fibroblasts, here color-coded. And the most interesting one was this particular one, which is here. This was the only one really driving myocarditis. It's an inflammatory forms of fibroblasts and express CXCL9, as you can see here. And this population also tracked with troponin, also tracked with levels of severity. So now I told you that there's CD8 T cells, there's specific dendritic cells, and there are fibroblasts that all track with severity. We asked, do they co-localize together in the heart? Can this niche be a therapeutic target? And so we did macroscopy imaging to validate this. On the left is control, on the right is myocarditis. We stained dendritic cells in blue, fibroblasts in pink, 6 on 9, 10, 11 in green, T cells in orange. And to make a long story short, we can find them co-localizing more in cases than in control. And we even had 2 cases that recover from myocarditis and these niches disappear. So now we want to ask, can we find mouse model that can recapitulate these niches so we can do test different drug targets and try to kind of map the antigen recognized by these T cells. And so there's a few takeaways here. We found different populations of cells that are up in the heart, and those that are up, very often they're down in the blood, which suggests trafficking. We found different correlates of severity that tracks with troponin. I mentioned a few, including the dendritic cells and the fibroblasts. And we identified some cell subset in the blood that tracks with fatality. And we showed that the biology between tumor and heart is distinct, which suggests an opportunity to identify drug targets that could decouple response from toxicity. Now with that, one of the aspirations is to be able to identify drug targets and help guide the next generation of clinical trial. And so while Kira's going to tell you more about the trial, these findings were done when the trial was already launched. It still supported the trial. So what we did is for every cell here we've identified in the heart, we looked at if they express certain drug targets currently in trial, whether it's checkpoint or drug used to treat myocarditis. And indeed, we could identify that the indirect target of the CTLA-4 immunoglobulin fusion protein abatacept was indeed expressed in the immune cells of the heart.
So just like she said, this is a global trial that is sponsored out of Mass General Brigham. So the lead PI is Dr. Tom Nealon and Dr. Paul Ritker at the Brigham. Our site PI is Dr. Dan Zlotov, and it's looking at abatacept. Just so, like she said, it's kind of a CTLA-4 agonist to put the checkpoint back on the immune system. And it's really because myocarditis, which happens about 1% of cases, can be fatal in upwards of over 30% or have a cardiac major adverse event. This study is looking at the normal steroids that we do plus abatacept on day 1, day 2, day 14 with an optional dose at day 28. The clinical outcomes are going to be incredibly important. As of this week, we've rolled in 144 patients. Again, these are rare patients across 48 sites in the US and Canada. To put it together, though, Mass General has enrolled 27, Brigham has enrolled 9. Together, our 2 institutions have made up 25% of the enrollment, showing that the structure and the patients that we care for are set up for research. In addition, we're collecting samples. So just like Chloe showed you 13 samples in that Nature paper, the reality is we have over 70 now, and we're collecting blood serially, as well as tumor and heart tissue.
So much more to do. I want to give you a really quick vignette on checkpoint nephritis, which is work we're doing together with Dr. Seiss, and that's co-led by Kavita Mistry, which is a nephrology fellow, and Isabella Kernan, who's a bioinformatician. Acute interstitial nephritis occurs in 2 to 5% of patients treated with checkpoint. The biopsies is the golden standard, but it remains risky and invasive, and we have the same type of problems in terms of treatment solutions. And so there's a key need for developing diagnostics. And just as a vignette, this is a project that also bridged between MGH and Brigham and Women's Hospital. They built a cohort of patients from which they collected urine specimens, including checkpoint nephritis, 57 cases, and 2 types of control, those that had non-institutional nephritis, AKI, and those that were treated with ICI but no stable kidney function. And they looked at secreted factors in the urine of these patients. And you heard about 6L9 a few times already in my talk. This was some of the lead chemokine identified in the kidney of these patients, as you can see here, much higher in checkpoint nephritis. And they went on to validate this with another technology, which is called mesoscale, and they use electrochemiluminescence to detect and quantify protein samples. You can actually quantify the level of it. And they validated this factor in the urine and could predict with very high accuracy if a patient had checkpoint nephritis just by measuring CXL9. Now, let it sink in. Imagine if we didn't have to do a kidney biopsy moving forward, and we could just measure one factor in the urine to diagnose. And so, after so many years, we're working on this. We're finally finding some initial targets. So I showed you how we're doing this using 3 different complementary strategies, focusing on the tissue, comparing with the tumor and the blood. The work co-led by Daniel Zotov, Steven Blum, Ramesh, Neil Smith, and Isabel Kernan was just published last year in Nature. We also published last year our work on colitis, also identifying some potential therapeutic targets, led by Molly Fisher-Thomas, who now has started her lab at OECHU, and Kamil Sławiekowski, computational biologist. But we have these ongoing effort using similar approaches as what I just described across the kidney, the liver, the joints, thyroid, neurotoxicity, and pneumonitis, and across a large translational team. You can see there's always at least 2, if not 3 people, if not more. That's because we have physician scientists coming together with bioinformaticians and immunologists doing this work. And by doing this work across all organ systems using the same framework, it means we can bring all the data together and start finally answering questions. Are all these toxicity created equally? Should you all be treating them the same way? Can we identify predictors for patient at risk? Can we identify the programs that are distinct from tumor response? That's the type of work we're doing. Ultimately, on the translational side, with the goal of defining these drug targets, we're using different type of data modalities to do so. So today I show you a lot of single-cell multi-omics, but we're now integrating, proteomics, function, and so forth to build these predictors. So with that, we're really proud of this grassroots. This is completely grassroots. This was not like top-down funded. It's the community coming together on the clinical side, on the translational side to set this up. We're crossing boundaries across 6 departments and 10 divisions of medicine to make this happen. We have multiple shots on translational goals because we're doing the same approach across all organ system. We are geared to succeed in identifying novel predictors and druggable targets, which can help inform the next generation of mechanism-based trials. We've also been training several fellows that are now faculty across different divisions of medicine and oncology that we're really proud of. Dr. Blum here just started his lab at MGH, joining this larger team effort. A lot of them have been funded through different fellowships, including our T32 programs. And this is one of our proudest achievements now.
We still have so much to do for patients. We feel like that was elementary school. So when we think about the opportunity as we come together at NGB Cancer Institute, an oncotoxicity program, so not necessarily just immunotherapy, but thinking about toxicity broadly. Dr. Nicole LeBluff over at the Brigham, as well as many subspecialists, have been coming to this table, sitting side by side scientists, because MGB Cancer Institute really has a chance for a pioneering initiative to reduce the burden of toxicities from all forms of cancer treatment. And it's this multidisciplinary approach that I hope you know is so unique. It's unparalleled to bring disease experts together and to leverage the resources that we have with our existing teams. And imagine if we could coordinate and really be comprehensive with our care, always conducting cutting edge research and training the future leaders in the field. The program will establish MGB as the cancer center that leads in not only life-saving, but quality of life-saving care and research across the entire cancer journey. And so to unify oncotoxicity care, research, and education to really transform outcomes, where are we headed? Retention is critical for these subspecialists. In addition, we've got to integrate the expertise and the intake to make their lives easier.
And that includes also training the next generation of clinical and translational leaders to make this happen.
We have to increase access and we have to scale this. Imagine, just like we have multidisciplinary cancer care, subspecialty multidisciplinary coordination could happen too. We could integrate more virtual visits. We could think about scaling the second opinion service nationally as well as globally. And high-risk clinics, we know that patients that weren't allowed in early clinical trials are now being treated. So how do we anticipate and mitigate complications? How do we set up co-management of these high-risk patients?
While at the same time continue to integrate sample collection so that we can create a roadmap for defining new drug targets.
And chronic toxicities matter. People are living, and they are a subset, almost 40% with chronic toxicity. So how do we advance survivorship research and long-term quality of life?
And in parallel, similarly, continue to integrate sample collection so we can build this biobank and create a roadmap to define better drug targets and biomarkers.
And registry integration, not just patients across the two sides of town, we have that. But how do we integrate it with patient-reported outcomes, with real metrics when we think about continuous learning? And we need to educate more broadly, even across our own system, but across New England and globally. And side by side, the key to this has been clinicians and clinical research sitting side by side the translational and basic scientists. So we have to focus on cutting-edge science being integrated.
And that includes going beyond what we started doing, right? Thinking about patient-derived models, expanding our genomics and proteomics profiling, thinking about bringing all of this through the lens of AI-driven data analysis so that we can nominate better biomarkers and target discovery to fuel the next generation of clinical trials. And so it takes a village to do this. I try to highlight some of the key people that did the work along the way. I couldn't do this without my awesome partner in crime, Carrie Reynolds, and all of the funders and the patients.
We really want to thank the patients and the families and every name on this. We are the ones presenting, but make no mistake, the reason why this is happening is because of the collaborative effort across the institutions. And we're going to leave the last word to Carol. So Carol was the 70-year-old breast cancer patient. Carol is sick 177 when you see all of those graphs and figures. These people all have a story.
Ms. Jensen, it's been such a pleasure to care for you. I'd love to just know when, if we thought about that long course of illness, what was it like at one of the darkest moments? And then how are you feeling now? And what's your day-to-day life like?
Well, my darkest moments, learning that I had cancer in the first place was a very dark moment. But when I had been treated with immunotherapy and all the complications came up, I was scared. Obviously, I was afraid, but I also had a sense of that the team knew what they were doing, and I felt cared for. I felt like I was being held in the hand of them. And now, having worked through all of the medications and several years, my life is so good. I'm 73 years old. I walk 1.5 miles every day. I'm able to care for myself completely and travel. So yeah, it's been a wonderful, not a good experience. It's not fun to have cancer, but the outcome has been worth everything that I went through.
And we are so excited too when we saw those most recent scans and all looks good with the cancer. And so that is the question, would you do it all over again?
And a New York second. I would never think twice about it because I know it works. And
in a New York second, so not even a New York minute, I really do feel like David would be proud of the team. I certainly am. We'll take questions.
there any question online? No question. Brian, do you have a question for us?
Yeah.
So it's amazing work, and the team is incredible. And I guess my the biggest question is, how will we be? Let's say the urine thing, right? I guess my question is around biomarkers. And you have a potential candidate biomarker. How are you going to ever validate that to a degree where you then can say this is the new way that we diagnose or help to diagnose nephritis caused by checkpoint inhibitors?
Well, we obviously need to validate this in quite large independent cohort. And so this is the work of Dr. Seiss, who's actually been working really hard of building prospective cohort so we can do a trial and assess this. But it's also important to validate beyond our course of MGB. So there's actually partnerships already being set at Yale and potentially beyond. There's always the challenge of making sure that this can happen. They're also trying to think about, without going into too much detail, and trying to find an easier way to measure this quickly to transform this as a diagnostic. So the short answer is, for everything we're discovering, whether it's the biomarkers for the heart or the factors in the urine, we need to continue biobanking samples systematically so that we can assess how valid these results are, first internally to help decrease our risk, and then through collaborations across institutions. And ideally, it's not just a diagnostic, right? You would want to know how early you can detect them. So there would be tremendous value in investing in biobanking specimens across cancer type as they come and visit the MGB cancer system, like you've done, Ryan, with the 11-1-1, where you follow all your cutaneous melanoma patients. If we could do this across all patients, we could achieve these goals.
And just to add, Ryan knows well, but we also have an alliance for the support and prevention of immune-related adverse events. So this was really kind of brought together with the Sick Service and MGH, as well as the Brigham, and now over 30 institutions globally. This is now part of ASCO. And I really do feel like if we start to bring everybody together and we think about non-invasive collection like urine, CSF, blood, this could be scalable at these other sites too, in a different way than, say, kidney biopsy, fresh, getting to Chloe across town 30 minutes or less, despite traffic at 5 o'clock in Boston. So the reality is, I think we do have to continue our collaborative efforts, both in the country and honestly globally. Thank you guys for being here.
Are there any other questions online? Well, Carrie, you answered my next question, which was going to talk about Aspire. So
I should say it's Aspire.
Yeah, exactly. So the alliance is Aspire. And I just want to say it really initiated with, how do you define a field? Okay, so just several years ago, I said, what are the ICD-10 codes for the immune-related adverse events? Because I kept putting like inflammatory bowel disease or autoimmune hepatitis or some other thing that I was borrowing from those. And the first drug was approved in 2011. Right. And so I thought, who are they? They're going to have to come up with the ICD-10 codes. And it became pretty clear that we might be they. And so it really was here where we started to define how different these are from their autoimmune counterparts. And we went in through the CDC and CMS to create ICD-10 codes. We learned a lot over an 18-month process. They are now in effect as of October of last year. But in order to get the public support that we needed, we started to think about toxicity efforts that might exist anywhere and everywhere. And that was the foundation. All of us championing the ICD-10 codes really led to the start of the alliance, which is Aspire. And then once we started meeting regularly, virtually 4 times a year, we meet for case conference 4 times a year. Anyone is open and welcome. We also have an in-person meeting. As we came together in this alliance, that gave us the power in the last year to go to ASCO to see, could we dock within the American Society of Clinical Oncology, which is one of the larger organizations. And so now that we've been adopted by ASCO, at our first annual meeting this past year. I really do think there's a way to continue raising awareness and hopefully, also continue to understand the need for funding, because research in this area is critical as more and more patients are treated.
That's awesome. And I liked your -1 where we got to sneak out in the back of the ASCO McCormick Center and ate Chicago deep dish pizza and had like a meeting and we got kicked out.
What he's talking about is the year before we were adopted by ASCO, we really wanted to be adopted. So I set up this meeting in the back of just the patio, and I had balloons. We had a sign for our Aspire Alliance. I was like really excited. We got about 50 people together from all these institutions, and then they started ripping our stuff. They're like, "You cannot meet here. This is not legal."
No, we need to talk about your ability to be covert sometime.
Yeah.
It's okay. Actually, I just want everybody to again acknowledge the wonderful work and beautiful presentation by Carrie and Chloe today. It's been such a pleasure to hear about what you're doing and what you're going to be doing in the years to come. Thanks.
Watch this.